Hello, everyone, and welcome. I'll start by introducing myself. My name is Dr. Noha El-Sarqa, and I'm a consultant medical microbiology at NHS Grampian. This is in Aberdeen, UK. And I'm an honorary clinical senior lecturer at the medical school at the University of Aberdeen. I'm also the vice president for communication for the Royal College of Pathologists. I'll be handing over to our webinar host, Joy, shortly, but I would just like to start by introducing the Royal College of Pathologists and giving you a bit of a background. We are a professional membership organization and have a charitable status. We have over 12,000 members who are mainly pathologists and scientists working in 17 different pathology specialties. We also have undergraduate members and foundation fellows, so get in touch with us if you would like to find out more about this. Most of our members work in hospitals and universities in the UK, although we also have international members working and training in a wide range of countries all around the world. We oversee training, exams, and professional standards across the pathology specialties. Our speaker today, Dr. Matt Clarke, is the college undergraduate and foundation lead. His role involves leading and coordinating the college's undergraduate and foundation activities with the aim of increasing awareness of the pathology specialties and inspiring the next generation of pathologists and clinical scientists. I'm delighted to welcome you to this webinar on neuropathology mystery cases. Now over to Joy.
Thank you so much, Dr. El-Sarqa. So good evening, everyone. Thank you so much for joining. I'm Joy, the outgoing president of the Imperial Pathology Society and the current vice chair of the Pathological Society Undergraduate Network. So alongside UCL and Manchester Pathology Societies, we've been supporting the college in running a series of careers webinars over the past couple of years. And we're delighted that Dr. Clarke has agreed to speak to us about some mystery cases that he has encountered in his role as a neuropathologist. As well as his role within the college, Dr. Clarke is a consultant neuropathologist and an NIHR clinical lecturer in diagnostic neuropathology based at Great Ormond Street Hospital for Children for his clinical work and the Institute of Cancer Research for his research. So Dr. Clarke will speak for around 45 minutes, and that will be followed by a Q&A in which Dr. Clarke will answer your questions. So please remember to post your questions for him in the Q&A box. And if you see any questions in the question panel that are similar to your questions, please upvote them instead of writing a new one so that those questions are answered first. So with that, now please welcome Dr. Matthew Clarke.
Thank you very much, Joy and Noha. Just going to share my screen. Lovely. Well, thank you very much for that lovely introduction. And it's a pleasure to be here tonight with you all. And thank you very much for joining for this talk. Yes, as has been mentioned, I'm a consultant pediatric neuropathologist at Great Ormond Street and clinician scientist, but only very recently. So I only just finished my training a couple of weeks ago. So very new to the role. But it's a pleasure to talk to you a little bit about my specialty tonight and some interesting cases that hopefully you will enjoy. But first of all, I thought I'd tell you a little bit about what neuropathology actually is. It's one of the 17 different pathology specialties, but probably one of the less known ones. We're actually quite a small specialty, and there are only currently 19 trainees across the UK in this specialty. So they're a very niche area of that. But our fundamental principles remain the same as the other specialties. So we are very much medical detectives trying to work out and diagnose different diseases of the central nervous system, muscle, and nerve in particular. We don't work on our own. We work with a whole team of different scientists, oncologists, neurosurgeons, and neurologists, all working towards the same aim. And the principle of our work is looking at the microscopic appearance of different tissues and fluids, looking at those features and trying to work out what may be wrong with a patient. And we have a whole range of different tests, some of which I'm going to show you a bit about tonight, to bring all these jigsaw pieces together and make an accurate diagnosis for our patients so they can get the right treatment that they need. And we're very much in the era of genomics at the moment. And this, instead of just looking down and looking at the microscope, looking at the microscopic images of the different tissues, we can now go inside those tissues, inside those cells, and look at the genetic material and proteins associated with them. And this is opening up a lot of avenues into understanding about the different mechanisms of disease. And again, I'll show you some examples of how this has really transformed our management of patients in neuropathology. And part of the problem with this is, and one of the challenges that we've had is that the different tumors can look very similar to each other, particularly brain tumors. And previously, we just have to make our best guess as to what the diagnosis might be. But these two tumors that I'm showing on the screen here now, these are probably, hopefully I can convince you they look very similar to each other. But the one on the left-hand side has just a single alteration in its DNA, and the one on the right-hand side has 10 different alterations. So although they look very similar, their genetics are very different to each other. And so they require different treatments. And so that's, again, the principles of molecular pathology helps us to identify those changes. And the reason why that's really important is because when we provide this information to our clinical colleagues, they need to know which drug strategy is going to be the most beneficial for these patients. And if they get onto the wrong pathway, their outcome is not going to be as positive for this as well. So there's a big role and a responsibility that we have to play with this. And I love this picture. This is it was designed based on the Andy Warhol soup artwork by my supervisor, Professor Chris Jones. And the principle is a bit if you imagine tumors as soup, but we know what soup is, but there are lots of different flavors and there are lots of different ingredients to these soups. And that's effectively what we do with brain tumors. We know what a brain tumor is, but we now need to go in to look at those ingredients and understand what that makeup of that tumor is to help us move forward in terms of our understanding of them, but also how we can successfully treat them as well. And before I go on to some of these cases, I just wanted to talk to you about some principles of molecular pathology that might help you to understand a bit more about some of these tests we do. And one of the things that was discovered a few years ago is that when you look at the DNA from any known brain tumor and look at where different methyl groups are added along the position of this, if you compare every known brain tumor, they each have a different profile of where these methyl groups are added, which represented a really exciting opportunity because if you can profile those methyl groups, and you may be able to, if you have a case that you're not able to diagnose accurately looking down the microscope, you could use this test to actually help you and support the diagnosis. And so this led to the development of the Heidelberg Brain Tumor Classifier. And so the methylation profiling is one of the fundamental principles we use in diagnostic practice now. And this picture on the right-hand side is a visual way of representing methylation data. And you can see that each type of tumor is represented by a different color, but each dot represents an individual tumor case. And you can see that tumors with a similar methylation profile cluster together in a tight cloud. And so when we get a case, we can sequence the DNA, look at where these methyl groups are added, and add it into a plot like this to see what type of diagnosis the classifier predicts it's going to be. And this has been really helpful because it can actually change the grade of different tumors. So some can go from what we think is low-grade tumors based on their microscopic appearance to high grade based on their molecular findings, and vice versa as well. So it really helps to tailor the treatment prospects for these patients. And what we get from the classifier is it gives us a score from 0 to 1. 1 meaning it fits exactly with this particular type of tumor, and 0 meaning it doesn't fit with any of the known cases within the classifier. And so we can use that score and that classification to help understand and to assess how reliable this test is in terms of our diagnosis. Before I go any further, we also need to look at what normal brain actually looks like down the microscope. And with any specialty, we have to understand what the normal is before we can look at the abnormal. And this is a bit of brain, a bit of cortex down the microscope. And just as a rule of thumb for tonight, anything that looks a dark purple color generally is a nucleus. And then you have the cytoplasm around the outside of it in a pink color. And just to orientate you to what we're looking at here, these cells that I'm circling around now, these are neurons that you expect to find lots of those within the brain. These smaller, darker ones are the astrocytes, which are the main structural components of the brain. You also have some little capillaries that are bringing the blood into the brain as well, importantly. And all of this sort of cotton wool pink material around, that is the neuropil, which are the different connections that connect all of these cells together and allow us to do our normal functions and allow the brain to work properly. So that's what we expect to see normally. So keep that image in mind when we move on to some of these cases. So let's have a go with this. So the first case is a child, a very young baby, actually, a child diagnosed before they were even born with a brain tumor. Now, shortly after they'd been born, they underwent a biopsy, and a sample of that was sent to the neuropathologist who diagnosed the tumor as a high-grade glioma, so a very aggressive type tumor, prognosis of around 2 years. And a few days later, the baby underwent further surgery to try and remove all of that tumor, but sadly, some of it was left behind, and a sample was sent to me to have a look at as well. And these are the MRI scans of the tumour before the operation. And you can see how extensive this is. Solid components of it, some that is cystic, occupying a significant proportion of this baby's brain. So a really, really nasty looking tumour that was actually actively growing as well. But after surgery, the surgeons had done a remarkable job, and I've outlined in red, this is just the remaining bit of tumour that was left behind. But the trouble is with an aggressive tumour, if that's left behind, it will continue to grow. And then, unfortunately, that will be a bad outcome for our little baby. Looking down the microscope, hopefully I can convince you, this looks very different to what we'd expect with normal brains. So these nuclei here, very dark in color, but also very variable in terms of their size and their shape. And that's what we call nuclear pleomorphism. Also, this is an area where a lot of the tumor cells have actually died, and that's also a marker of how aggressive this particular tumor is. And again, another example here, a really gigantic nucleus that's about probably 4 or 5 times the size of its nearest neighbor here. But also we start to see other cells that are in the process of dividing, which again is a sign that this is a fast-growing and aggressive type tumor. As I mentioned, DNA methylation is one of the key parts of this work and understanding a little bit more about these brain tumors. And we also know that methyl groups tend to be added around the regions of promoters of particular genes as well. And so if the promoter region is actually methylated, the gene tends to be switched off. And we see this a lot within tumors as well. And it influences the type of profile that we see in terms of the gene expression of these tumors. So what we did subsequently, we diagnosed this as a high-grade glioma, and then I decided to do some DNA methylation profiling to see actually what type of tumor it was. But we got some very interesting findings because this is a picture showing, again, just to remind you, these clouds represent all the different high-grade glioma types represented by different colors. And my case actually landed in this circled area here, a group of tumors that we didn't really know a lot about at the time, a very new and novel type of tumor now known as the infant type hemispheric glioma. And so again, as part of my research work and my PhD project, I tried to understand a little bit more about these and also more about this little girl's tumor as well. So molecular pathology was really helped to save the day in this case, because when we did some further sequencing of the DNA, we identified a fusion. And so when I looked at chromosome 12 and chromosome 15, there was a break at this point on chromosome 12 and a break at this point on chromosome 15. And the two bits had joined together, which meant that now the ETB6 gene that was normally on chromosome 12 was next to the NTRK3 gene that was normally on chromosome 15. And that ATP6 gene was overactivating the NTRK3 gene, and that was what was causing the development of the tumor. Another molecular test we can do to help confirm this is something called fluorescence in situ hybridization. So I could use fluorescent labels of these two genes to see if they were in fact fused. So I labeled the ETB6 gene with a green fluorescent marker and the NTRK3 gene with a red fluorescent marker. And usually, because they're on different chromosomes, just like in this normal cell here, they should be far apart from each other because they're on separate chromosomes. But where you see the yellow arrows, the red and the green signal have joined together, indicating that these two genes have fused, confirming and validating our results. So this was really interesting. And this can happen, this type of mutation can happen across all different cancers, not just in brain tumors, including lymphomas and other in bowel cancers, lung cancers as well. But it's a really important thing to understand and pick up from your tests because there are potentially things that you can do about it. So the conclusion for this case was that this baby had an infant-type hemispheric glioma with an ETV6 NTRK3 fusion. And the reason why this was exciting and interesting was because there is a drug that targets this particular fusion. And it avoided her having to have damaging radiotherapy and chemotherapy at such a very young age. And so I then went a step further to try and see whether this would actually work clinically. So one of the things I also did in my project was to go to theatre and collect some tumour samples at the time of operation, bring them back to the lab, and then I could grow them in these culture flasks. And these are some pictures of some of those cells that are actually growing on the bottom of the flask here in this dark red type media. And that allowed me to actually do some special tests. So I could do some testing to see whether the cells were actually sensitive to these specific drugs. And I got some positive results to show that in fact they were sensitive and that the drug was actually killing these tumor cells. So that was a really important translational work. So what happened to our little baby then? Well, after she got the result from me that she had this fusion, the oncologist started her on the drug crizotinib, and you can see that the tumor after the first MRI scan had shrunk. And then she was changed to another drug called larotrectinib. And I'm delighted to say that she's now alive and well at 7 years old. And after originally having a prognosis of 2 years, she's at school and doing really well as a result of it. And so, again, a really nice example now of how neuropathology has actually really helped save this little baby's life, but also many others around the world in a similar situation as well. So we can really make a difference. So case number two, this is a 54-year-old man who's had a long-standing history of a left frontal cystic tumor. So it's been there for quite a while. The team have been keeping an eye on it and just seeing what sort of behavior it's imaging. And sometimes that's the case, really. We don't always dive straight in to remove something, and we want to see it biologically, how it's behaving. But the imaging had shown really large area of calcification with it. And that's actually something we can see with a lot of brain tumors. But eventually a decision was made by the multidisciplinary team consisting of neuropathologists, radiologists, neurosurgeons, neuro-oncologists to take the tumor out. And this is what we were sent to the lab. So two fairly large bits of tissue, but you can see quite a lot of different patterns in terms of the pink and purple staining here. So let's have a look and see what we've actually got. Well, it looks a little bit of a mess here, actually, but all this dark purple wave-like appearance is actually calcium. So there's lots of calcium within this particular tumor. But also there are other areas where you can see lots of vessels here with red blood cells and also quite a few inflammatory cells around the outside of them as well. So two different components here. And again, just another area just showing you these large areas of calcium. It was all over the place, but also some reactive type astrocytes that look very plump and big compared to that original microscopic image I showed you at the beginning, where they were very much smaller and much darker in their appearance. So they're reacting to something present here as well. Another area here, you can see this large area of lymphocytes, so lots of inflammation happening associated with this lesion as well. And again, another example of one of these plump astrocytes. And then when you get your eye in, you can see that there are quite a few in the adjacent brain as well. So something reactive happening associated with this. And then another bit, which looks exactly like bone. So this is the sort of lamellated bone we normally see, and you can see here as well these little osteocytes present within this. So a very odd mixture of things that we've got within this. I did some molecular testing, and the brain tumor classifier didn't classify this as anything. It didn't give us a reliable score at all. And when looking at the copy number profile, when we're dealing with a tumor, we see lots of gains and losses of particular chromosomes or part of chromosomes. So you can see the chromosomes labeled along the bottom here, and all of these dots are here are concentrated around the 0 mark. If there'd been gains of chromosomes, we would see all the dots up here, or if there have been losses, they would be all down at the bottom. But this is not showing any changes at all, which is a marker, in fact, of normal tissue. So what actually was this? Well, it was very well, just to summarize what we got, it was a very well demarcated lesion consisting of connective tissue, extensive calcification, bone formation, and patches of inflammatory cells with a reactive gliotic wall around the outside. So the diagnosis was in fact something called a calcifying pseudoneoplasm of the neuroaxis, or a CAPNON. I don't expect any of you have ever heard of that before. This is actually quite a rare condition, and it's otherwise known as a brain stone. And these are quite rare. They, again, present in patients of any age, and they can occur both within the brain and the spinal cord, but they're not actually thought to be a tumor. They are more a mixture of different tissues, that what we call a hamartoma, where they're very disorganized and they've not formed in the right way, and so give you this mixture of different things in the component of them. So they don't actually necessarily do any harm, but most of them end up being resected because of the associated symptoms of them. But yes, an example of quite a striking one. I've only ever diagnosed one in my entire career, of a brainstone that I thought you might be interested to see about. So these are just a couple of papers if anybody would like to read a little bit more about those in your spare time. So the third case, this is an 11-year-old boy who presented with cranial nerve palsies and ataxia. So this could be examples of where there were abnormal eye movements. It may be where there's weakness in some of the facial muscles. He also had some motor deficit as well, so perhaps some weakness along one side. And he underwent an MRI scan that picked up a lesion that the surgeons wanted to biopsy. So if we're looking at this MRI scan here, I'll just direct you to the brainstem, because there's this large lesion here that's actually causing an expansion of the pontine region. So the pons is a lot bigger than it should be. And it's very difficult to actually make out the other structures of the medulla and the midbrain here as well. So something that's causing pressure effect here. And also you can see the cerebellum, it's pressing on that outside portion of this also. So the surgeons did a biopsy. You might ask, well, why didn't they resect that? Well, the trouble is, because of its location in the brainstem, there are really important nerve and structures there which are responsible for cardiac and respiratory function. And so if you intervene too much in that area, you're going to leave the patient with a significant number of problems. So the best we can do at this stage is to get a biopsy of it and then hope that we can use chemotherapy and radiotherapy to treat it. But again, you might recognize some of the features we saw with the other tumor, very pleomorphic looking nuclei, very large size in terms of the nuclei as well. So this one is about 5 times the size of its neighbor as well. And also something I don't like to see as well with these tumors, lots of mitotic activity. So again, showing it's potentially a high grade tumor and it's dividing a lot, even in just this small view of this tumor here as well. So what is it? Well, there are some stains that we can actually do to identify the molecular features of it. And one of them is detecting a mutant protein that we call H3K27M. And it's a mutation in one of the histone genes. And the histones are some of the proteins that DNA is wrapped around to package it up into chromosomes. And when we look at this stain here, the cells, the tumor cells, are all lighting up brown with it, indicating this mutant protein is present. And the effect this mutant protein has is it interferes in the function of an enzyme that normally causes methyl groups to be added onto that histone gene. And so when we stain for these methyl groups with this stain, the H3K27ME3, you can see that the tumor cells are actually remaining blue. They don't actually light up brown with them. And in fact, the cells that are brown are the normal ones in the background, so showing how extensively infiltrating this tumor actually is. Another stain that we can do, it stains up the filaments, the neural tissue in between. And again, you can see these lovely strings of neural tissue and also how infiltrating this tumor is through all of this tissue and how challenging this is when it's present within the pons. So once again, I can do DNA methylation profiling to confirm my diagnosis because there is a group of tumors that are characterized by this K27M mutation. And so let's see where our case lands. Well, straight in the middle of that cloud and with a high calibrated score confirming the diagnosis. So this sadly is a high-grade tumor, a WHO grade 4. That's the maximum we can give it. And it's called a diffuse midline glioma. So a little bit more about these. These are tumors that are characterized most frequently within children, but we do see them within adults, but they are almost always located within the brainstem or the midline of the brain. So it's a tumor that has a molecular characteristic and specifies where the tumor is going to be located. We never see them in the cerebral hemispheres, always in the midline structures as well, but sadly, they have a very, very poor prognosis. They do, as I mentioned, occur in the adult population. So I think the oldest one I've diagnosed is a patient who is 72 years old, but we can also get some other genetic alterations associated with them as well. So sadly, these have a very, very poor prognosis, but in some cases, we can do something to help with these. And again, molecular pathology can help us. This is another example of one of these tumors that was present within the brainstem. And looks a little bit different to what we saw previously. So there's a bit of hyaline material here. The cells have got a little bit more what we call spindle morphology. So they're quite oval and elongated as well. And there are also some bits where the stroma is almost falling apart. And this is a very characteristic type picture we see with some low-grade glial tumors. There's also, you may recognize a little bit of calcium spotted around as well, and a bit more as where the stroma has become quite hyalinized over here as well. So a bit of a mixture of different patterns associated with it, but also some features that we normally associate with high-grade tumors. So this is a bit of what we call microvascular proliferation, where the blood vessels have actually divided to increase the supply of blood to the tumor cells so they get all the nutrition and the oxygen, etc., that they need to survive and grow. Now, when I did my immunohistochemistry for this one, again, it confirmed that this had this horrible K27M mutation associated with it. And again, this is Ki67, which is showing how many of those cells are actually dividing. So again, showing it's a high-grade tumor. But what it also showed was that there was a BRAF V600E mutation present within it. This is quite rare, but some of these tumors do contain this. And this is really important to identify. So again, I did the methylation profiling on this, and we got a nice score of 0.955, so really close to 1, confirming it's one of these diffuse midline gliomas. The copy number profile, again, didn't really show a lot going on, but again, the sequencing I did confirmed the presence of this BRAF V600E alteration. And the reason that's important is because that is targetable. And so this patient could now be started on either a MEK or a BRAF inhibitor. And the studies that have been done so far have shown that these patients can actually be long-term survivors with it, living for many years or so with their tumor under control potentially as well. So a small subset of these patients, we can offer some potential treatment that's going to help improve their prognosis. So again, molecular testing in neuropathology has really helped to identify these cases. Moving on to case number 4, this is a 51-year-old female patient who presented with headaches and nausea, and it's been gradually getting worse over the last 3 weeks. She went to an optician's for an routine appointment, and the optician noted papilledema at the back of the eye, and this is what it looks like. So normally you should see a nice crisp outline of the optic disc, but here you've got quite a lot of blurring, and this is because of edema that's being transmitted from raised pressure in the brain down the nerve, and it's blurring the outline of the disc. And so opticians are trained when they see this needs to be referred, and a doctor needs to have a look at the patient urgently. And so they were referred for a review. And subsequently, they underwent an MRI scan. And here you can again see that it was how right they were to refer the patient. So there's a large lesion here that's present, but also there's all this white stuff here is edema. So a lot of fluid reaction associated with this lesion, likely to be a tumor, but also there are solid bits to it. There are also other bits that are more fluid-like as well. And it's also causing significant shift in the midline, so causing a pressure effect, blocking part of the ventricle over here, and again showing how much pressure is in this confined space of the skull, and why we were seeing that change in the eyes as well. Now, one of the things that we also do in neuropathology is if we're not really sure, based on the imaging, what the diagnosis might be, and that's what we discuss in our MDT meetings, the surgeons may ask us to do an intraoperative assessment. So when they're in the operation, they will send a small sample of the tissue, rush it to the lab. We put the piece of tissue on a slide and smear it across the slide, add a stain called toluidine blue. And so within 5 minutes, we can put it on the microscope and see what we think the diagnosis is. And then we give the surgeons a ring on their tannoy in the theatre and tell them what we find and what we think it is. And that may then guide what they do with the operation. In some tumours, they may stop the operation altogether because they may think the surgery is not needed because they can use chemotherapy to treat it. Others, they will go hunting for as much of the tumour as they possibly can to resect it. So again, a lot of pressure on us in 10 minutes to actually give them the right answer for this. So what are we actually looking at here? Well, these, again, look quite unusual cells. So here we've got one cell that's actually got 2 nuclei, and it's also got a really prominent nucleolus within the center of it. And when you get your eye in, you can see there are quite a lot of these cells all over the place, but also some mitoses present within it as well. So it looks like an aggressive tumor. Now, the next day when I got the H&E slides, the striking thing about this was all of this brown pigmentation that was present throughout it. This was everywhere, and this is very unusual for a tumor within the brain. And again, looking a bit more closely, you start to see this pattern of the nucleus with the prominent nucleolus present here. Here's a mitotic figure. So it's all matching with what we saw with the intraoperative assessment. So what could this actually be? Well, when we see brown pigment in the brain, there are several things we can think of. It could be melanin. It could be hemosiderin, secondary to breakdown of blood products, or it also could be lipofuscin, which is a deposit we get within cells as we age. But in this case, given the cytology and the appearance of these cells, something melanin related was what I was thinking of. And could this in fact be a melanoma? So then I did some stains. And we have 3 markers that can tell us whether or not this is a melanoma or not. There's S100, HMB45, and melan-A. And you can see in all 3 cases, the tumor cells are lighting up brown, confirming that this is in fact a melanoma. But also, again, BRAF V600E was positive associated with this. So we've got a target potentially for this patient. So this is, although this is a melanoma, and we need to also decide, is this one that's actually grown within the brain itself? Because we have melanocytes on the surface in our meninges, or is it one that's actually spread from somewhere else in the body? So has the patient got a skin tumor that's actually metastasized to the location? So when we give this information to the team, they decided that the best thing they wanted to do, based on the imaging, was they wanted to send a CSF sample to see if there were any tumor cells that might have migrated elsewhere down perhaps the spinal column. And so that's the other thing we do as neuropathologists. We look at CSF samples. And here you can see these quite large cells with, again, very prominent nucleus. You can even see the detail, the little blebbing on the surface of them. Here, one's got 2 nuclei. And just to give you an orientation in size, this is a little lymphocyte that's nearby. So they're really gigantic compared to those. So what are these doing in the CSF? They shouldn't be there. The CSF should be completely acellular. So it's quite concerning when we're seeing things like this. And again, more of them here. This is one with 2 nuclei present, and another one down here as well. And also these ones in the background, these are macrophages and lymphocytes as well. So there's some inflammation responding to something that's going on within the spinal column. And again, another close-up, just giving you this, again, fine detail of these cells with this blebbing along the surface of them also. So I could do my stains again to actually confirm whether or not these were melanoma cells, and they lit up for HMB45 quite nicely here, and also for S100. So unfortunately for this patient, that meant that it showed that the cells had actually spread within the CSF sample and potentially could metastasize elsewhere within this. But because they were BRAF V600E mutant, that meant that she could be started on a BRAF and MEK inhibitor to try and treat this successfully. It's not necessarily going to be able to cure her, but at least it will treat some of the lesions and actually help to improve the prognosis for her. And because of our diagnosis of this as well, she was actually referred to the dermatology team, who actually identified a skin lesion that was very suspicious, that was later actually confirmed to be a melanoma. So it was one that had in fact spread to the brain from a skin lesion as well. So again, we can be quite helpful in terms of determining where the tumors have actually come from in neuropathology also. So case number 5, this is a 72-year-old man who presented with confusion and headaches, clinical course of a few-week duration with some nausea as well. And he was referred for an MRI scan, which again showed this rather nondescript lesion that's a bit difficult to outline. There's a bit of fluid around the outside of it, but it's quite non-specific. It's difficult to sort of outline where it begins and ends. So again, the surgeons wanted to get an intraoperative assessment for it. So we can see here little vessels, but there are also lots of cells that are sort of clinging around these vessels, and there are lots of little tiny ones present. And looking in a bit more detail at them, they're quite rounded and circular. So again, when I see this, it makes me think of inflammatory type cells or lymphocytes. So again, we get the H&E slide and we can confirm what we were suspecting. So again, lots of inflammatory type cells here. So when I start thinking about this, 2 things are in my mind. Is this an infection or is this a lymphoma? So a type of a tumor that's affecting basically the inflammatory cells. And again, further details here. What's making me think more about tumor is there's several mitoses present within this as well. So what the most common type of lymphoma we see in the brain is what we call a B-cell lymphoma. And B cells light up for CD20. And so when I did the stain for this, though, you can see just on the left-hand side, this is something we call glial acidic fibrillary protein. And this lights up the astrocytes that are in the background here. You can see some of the glial cells. But the CD20 is completely negative. So there aren't any B cells here at all, which was a bit of a surprise. But when I did the marker for a T cell, which is CD3, they all lit up for this. So it was very, very unusual to find this. And also when I did the proliferation marker, you can see they're all lighting up for this as well. So they were all busy dividing, indicating that this in fact is a lymphoma, but not the one I was expecting and not the most common. In fact, a very rare one, a T cell lymphoma present within the brain. And it's very, very rare to actually find that. So a lot of the time they actually present with lymphadenopathy in other locations of the body, but there are also some other molecular features. So we can do some different genetic tests to identify some mutations in particular genes like TP53, and also PCR that identifies this particular monoclonal population of T cells specific for this. But this is important because that patient can now go on to a treatment and chemotherapy, which will hopefully lead to a more positive outcome for them. Before I move on to the next case, I just want to talk to you a little bit about brain banks, and that's something else that neuropathologists are really involved in. And there is a really increased frequency of neurodegenerative diseases, lots of incidences of dementia that you'll hear about and see. And so understanding a bit more about these conditions is really vital. And patients are remarkable in that when they are diagnosed with a condition like this, many of them will actually consent to have their brain donated after they die to a brain bank to allow further research and study to happen to understand more about it. And so neuropathologists are involved in actually sampling those brains and examining them as well. And this is an example of what a normal brain looks like, just to orientate you. So here we've got the frontal lobe, we've got the parietal lobe, we've got the occipital lobe, the temporal lobe, and also the cerebellum at the back. And we've got over the surface this sort of grey-like material is the meninges, and you can see the blood vessels present here as well. And all of these folds of the brain are called the gyri. And that's effectively what we like to see. That's what a normal brain should look like. But of course, when we have a neurodegenerative disease, things can look very different to that. So this is the history of a 65-year-old man who presented with cognitive decline and dementia from the age of 60. He died sadly of pneumonia, but donated his brain for research to understand more about it. And the brain weight when we received it was 925 grams. And normally we would expect a brain to be upwards of 1,200 grams. So significant loss in weight. And apologies for the graininess of this picture, but what hopefully you'll see is where those folds are, they're really reduced in size, meaning that the gaps between them have really exaggerated. And you can see that's quite remarkable in the frontal lobe and also in the temporal lobe of this brain. It's not as badly affecting the posterior aspects of it as well, but this is quite a striking image where there's been degeneration of this patient's brain. And then looking inside it, you can see the ventricles are really very expanded. This is the caudate nucleus, this is the putamen, and this is the thalamus on this side. And also there's a big hole that's formed as a result of expansion of the ventricles in the temporal lobes as well. And the reason for that is there's less pressure on the outside because there's less material from the brain. And so the CSF expands the ventricles and makes them look bigger than they should be. And again, as we go further back through the brain, these ventricles are really dilated. And again, it's because there's significant loss of tissue from the outer surface of these lobes. So what is this? Well, so we've got quite a significant loss of tissue and we've got expansion of the ventricles. So the diagnosis for this patient is a frontotemporal lobar degeneration or an FTLD type dementia. But we can make that diagnosis or suspect that diagnosis just by looking at the brain, but we also need to look at it microscopically as well. And when what we look for is these things. So this is a inclusion within one of the neurons, and we can light it up with a stain called for tau, and it's a tau inclusion, a neurofibrillary tangle that characterizes this. So it shows that the degenerative change is happening as a result of this abnormal accumulation within these neurons. The presence of this protein is causing the damage to the neuronal function, and it's causing them to die, and that's what's causing less and less of the tissue to be apparent within the brain. So when we do this, it's really important to correlate what we see with our eyes with what we're seeing down the microscope. And sometimes we can see other patterns of this type of protein as well. So these are what we call starry astrocytes. It's the same protein, but lighting up in a different fashion with the brain. And this is a condition called progressive supranuclear palsy. And so just the pattern of the distribution of this protein can give us a more accurate diagnosis as to what it is. And so those samples, we can confirm what the diagnosis is, confirm that with the family as well, and let them know about it, but then also help to supply research material for further research. Another case is a 48-year-old female patient who presented with some quite odd behavior. The family noted a very rapid deterioration in her memory, and she was reviewed by a neurologist who noted myoclonus, which is effectively very sudden abnormal jerky movements, but also ataxia, so very sort of clumsy-like ability with problems with balance as well. But the classic thing with this is that she deteriorated very rapidly and died after only 2 years after the first presenting with these symptoms. So this raises really big alarm bells when someone has deteriorated so rapidly with a cognitive decline, but also things like myoclonus and ataxia as well. And the brain looked something like this. So again, you can see these large gaps present within, particularly within the frontal lobes here, and narrowing of these little folds on the surface. So showing that there's quite a significant loss of tissue associated with it. But what was the case? What was causing it in this case? Well, the fact that it was so rapid gave us a big clue to that. So this is the normal looking brain on the right, on the left-hand side here. You can see the neurons present here, how quite distinct they actually are, and also the astrocytes, like I showed you in the first picture. This is our patient's brain. And the first thing to note is we're not seeing these neurons. A lot of them have disappeared, but also there are all these white holes that are present within it as well. So there's almost like gaps. Someone's been punched holes present within the tissue as well. So this is quite a characteristic appearance, or what we call a spongiform-like change to the brain. But what it was actually caused by is prion protein. And this is what we should see normally, and this is what we were seeing in our patient's brain. And it was an abnormal folding of this particular prion protein that was causing a degeneration of the neurons and a degeneration of this patient's brain. And it's characterized by this rapid deterioration as well. So the diagnosis was something called Creutzfeldt-Jakob disease. It's a prion disease, which is a little protein, but it's almost regarded as an infection because it can be spread. The prion itself is triggered to misfold, and it's this misfolded protein that can actually cause the degeneration of the neurons, but it can also induce other prion proteins to misfold as well. So it has a cumulative effect and can spread throughout the brain that way. It classically causes dementia, but once symptoms begin, you can get very rapid deterioration for it. So whenever we have a case that's suspected of prion disease, we as neuropathologists have to take extra precautions with this and do it in do the post-mortem examination in specialized labs, protective equipment, similar to what we would do with a high-risk type autopsy. So because the risk is, of course, that we could potentially contract this and develop that as well. So we have to be very careful with these conditions, but fortunately, they're very rare. So another thing I want to, before I talk about the next case, I just want to introduce a type of tumor called a subappendimal giant cell astrocytoma, or best remembered and easily remembered as SAGA. And the reason I wanted to show you it, you can see these very large cells here with very prominent nuclei, quite atypical in their shape, but actually they're very low grade. These are grade 1 tumors, behave very well. And if you can resect them, the patient will do very nicely as a result of that. But they occur secondary to a tumor syndrome called tuberous sclerosis, and mutations in either TSC1 gene or the TSC2 gene. So remember what those cells look like, and I'm now going to show you another case. This was an 11-year-old patient who had a history of headaches and nausea, and his MRI showed he had a right occipital lesion right at the very back of the brain. And just to show you this quickly here, you can see it located down the bottom there, and it looks very high grade and aggressive. But when I looked at the histology, I got quite a big shock, because it had lots of cells that looked typical for a high-grade glioma, but it also had these very unusual, bizarre-shaped cells that made me think about what we see associated with a SAGA. And again, just another view over here, they're very almost bizarre. This one looks a bit like a butterfly in places. You can see mitotic activity as well, but they're scattered throughout this tumor on a background of other cells that look very different. So what I did some further sequencing of this, and what it told me was in fact that there were a series of mutations in the TSC1 gene, but only a very small number of them. And so what the hypothesis is that these cells are actually forming that shape because of this particular mutation within them. So now, because of the presence of these, that patient could potentially be put on a particular inhibitor that actually targets this gene and potentially could kill those cells off as well. But what's really interesting, and why I'm wanting to show you this case, is it shows you that in fact the morphology, or how those cells look, is actually influenced by their genetic alterations that are actually undergoing that. And I think in years to come, neuropathologists will be involved in a lot more cases that are very similar to this, where we're almost mapping on the tumor where the particular mutations are within the particular cells as well. Just a couple more cases to go. So this is a 60-year-old patient. She presented with nausea and some difficulties balancing, and the MRI showed a cerebellar tumor that the surgeons resected. And when we looked at it down the microscope, very almost artistic looking tumor, so cells that were full of little bubbles almost. And this is typical for something we call a Haemangioblastoma, and it lit up with inhibin, which is a marker for that. And so the diagnosis was a Haemangioblastoma, CNS grade 1. So why am I showing you this? Well, a few years later, she came back and the tumor had grown back, and the surgeons therefore resected it. But this is what the tumor looked like, as we would expect a Haemangioblastoma to look like. But there was a nodule here that looked very unusual and looked quite distinct. So when I looked down the microscope, this is what the Haemangioblastoma looked like. So fairly typical. It looked a little bit unusual in that there weren't so many vacuolated cells, but this would pass for one of those tumours. But this is where the nodule came in, and it looks a bit different. So we don't have the vacuoles here. We just have lots of clear cytoplasm. So again, very different to the original tumour. And I started doing a few stains, and you can see that this nodule doesn't stain for the CD56 stain. And also, again, a bit close up, it again confirms that. The other tumor bit of the tumor stained for the inhibin, which is the marker for the hemangioblastoma, but the bit in the middle was negative for it. So it almost looked like there were two tumors here. And again, I did another stain for CD10 that gave me the answer. And the answer was that this patient had a metastatic clear cell renal cell carcinoma that had spread into the hemangioblastoma and was actually growing within the center of it. And the patient we understood had a long-standing history of something called von Hippel-Lindau disease, which is what can cause hemangioblastomas. But what hadn't been mentioned to us was that she previously had a renal cell carcinoma resected about 15 years ago. And so no one had thought to mention that because it was so long ago. But in fact, those cells had survived that long and spread into that particular location. And so that was the confirmed diagnosis. But thankfully, she's done very well as a result of that. And hemangioblastoma, like I said, it can occur as a result of a mutation in the VHL gene. They tend to have quite a yellow color during the operation and can bleed quite a lot as well, so it can be quite challenging to resect. The von Hippel-Lindau disease, it's actually important for us to identify these tumors because they have a genetic inheritance. So they're autosomal dominant. So if we identify one, we can recommend that the patient may want to go undergo further testing to see if other family members may be at risk of developing it or certainly be screened for it. And that, again, is a really important role for the neuropathologist. And there are rare, very rare case reports where similar things have happened, where metastatic renal cell carcinoma has turned up within hemangioblastomas as well. So the final case for this evening has again been a bit of a puzzle. This is a 16-year-old patient who presented with a seizure. He was suffering with headaches, but also on the MRI, he had a right temporal lobe tumor that the surgeons again resected. They were a bit unsure about the grading of it, but they thought it was best to actually take it out. And this is the sample that we received. So a few pieces and fragments of tumor. But the two bits I want to draw your attention to are these bits. And the reason were because, again, I got a surprise when I looked at them. So they look very different to each other. So on the left-hand side, they're very oval and spindled. And on the right-hand side, they have what we call this fried egg appearance. So you've got the nucleus within the center and lots of white around the outside of them. So big sheets of them, but very different. I started doing a few stains as well and saw that one stain would light up one area, but wouldn't the other area. And again, I did another stain and got the exact opposite. So it was lighting up some cells on one and not on the other, which again was a bit of a puzzle. So I decided to do some molecular testing on both areas separately, even though they were from the same area. The methylation classifier told me that they were two different tumors, one called a pleomorphic xanthorastrocytoma and one called a glioneuronal tumor. Both of them detected a NTRK fusion, so something that's great that we can target, which was exciting. And the copy number chain profiles of both areas showed some changes. So both of them showed a gain of a gene called TERT. The one on the left-hand side showed a change called a chromothripsis of chromosome 4. So the chromosome had actually completely shattered, but you'll note it's not present on the other area. And also we've got a deletion of a gene called CDKN2AB on the area on the left, but not on the right. So how can we actually explain this? Well, it was concluded that this is a glial glioneuronal tumor with an NTRK fusion, and we discussed the possibility, could this in fact be two tumors happening in the same patient? But what's more likely to happen is that this is an example of tumor heterogeneity. So there are different areas within the tumor, and what's probably happened is a genetic alteration has happened in two different cell types, and then they have both. This has led to the development of two different tumors that are along different lineages. The good thing for this patient, though, irrespective of this, is that they can be started on that larotrectinib, that TRK inhibitor, to hopefully resolve the tumor and make it melt away as well. So that, again, was just a lesson to us. Always look out for different areas of tumors and don't be afraid to do some molecular testing because you never know what you might find. So to summarize, that was a whistle-stop tour of some intriguing and hopefully unusual cases that you might have found interesting that have been quite challenging to work out. But in a lot of cases, we can actually make a positive outcome for our patients. Neuropathology is a career filled with a lot of variety and a lot of diversity, and you never quite know, as you can experience from these cases, what you're going to see on your digital screen or your microscope. We integrate and engage with clinical team and management very closely. Medical detective work is the fundamental of what we do, and we bring all the jigsaw pieces together, as I've shown you today, with a variety of different conditions. It's a very fast changing and rapidly developing specialty as well. And there are extensive opportunities for engagement with teaching and research, which is what I've made a real fundamental part of my career with it as well. And you never stop learning. Even as a consultant, I'm learning something new every week with these new cases as well. So there are many questions left to answer. And if anybody ever wants to contact me to ask more about the specialty or anything I've talked about tonight, I'm very happy to be contacted outside of this webinar as well. So thank you very much for listening.
Great. Thank you so, so much for such an intriguing talk, Dr. Clarke. I've got a few questions. I am aware of the time, so we might not be able to answer all of them. But if for those of you whose answers are not answered, if those of you whose questions are not answered in this webinar, please feel free to email Dr. Clarke, because he was very generous and happy to share his email. So the first questions we have is actually around careers. So any tips for histopathology ST1 applications? These would be very much welcomed.
So yeah, as a neuropathologist, we all go through ST1. We have to go in histopathology for 2 years, 2.5 years before we can enter neuropathology. So getting some placements would be really good. Joining webinars like this is fantastic because again shows your engagement with it. My advice would be to go and visit your local, your histopathology department and speak to some of the registrars there because they will give you some fantastic advice and maybe even get you involved in little projects and show you some cases. What they're looking for from your application in your interview is someone who's really enthusiastic, someone who's interested in the specialty and has gone to the efforts to learn about what we do in the role. So that's some brief things, but very happy to talk to you outside of the meeting if you'd like some further advice about it.
Thank you. The next question is, interestingly, I recently had a patient on a stroke ward who was admitted with stroke and turned out to have CJD in the end. Are prions present in CSF? And is this how CJD is diagnosed? Thank you.
Very good question. Yes, so the importance of diagnosing this has been recognized. And so there are now different tests that you can do to pick that up and also other neurodegenerative conditions from the different proteins that might be deposited within the CSF, a bit like what we hear a lot about with blood tests for detecting tumors. And so there are becoming more remarkable tests to be able to pick that up. And again, just like with this patient example, where you may be suspecting another diagnosis, just getting a sample of fluid maybe provide you in the right direction and completely change your treatment trajectory for that patient as well. So yes, there are now things we're doing a lot more with CSF now to pick up those different proteins.
Great. And the next question is by Erica. And when a histopathologist decide to do molecular testing, what part of that do they take part in? Either library prep, sequencing, bioinformatics pipeline, or which part of the analysis is their responsibility?
Very good question. So it depends. As a researcher, I used to do all that myself, and so I've got a good background in understanding those tests. But when you're working on the clinical side, we have a whole team of people. We can't do all that on our own. So there are biomedical scientists, clinical scientists that help us with our analysis. But we work together as a team to select the right bit of tissue, the right area that we want to look at, and then we bring those results together and integrate them into our report. So it's a team effort where a lot of people do various different parts of it. So perhaps the neuropathologist doesn't necessarily do a lot of the practical work of the extraction and the library prep, but they certainly are very much involved in the selection of tissue, deciding on what's appropriate test, and then the analysis of it afterwards.
Great. So next question is by Aisha. Can you tell us what genes are turned off during methylation of tumor?
Very good question, Aisha. It varies according to which tumor you have, which is what's exciting about it and why we get different profiles for each of those brain tumors. So every single one that we know about, looking at perhaps 180 different types of tumor, has a different profile of where these methyl chemical tags are added. So wherever they're added, different genes are affected by it. And so to understand more about these tumors now, researchers are focusing on those genes that are switched on and off and see whether that actually influences, or is there something that we can do to actually help the patients from that. One of our big challenges in neuropathology is we're able to really define and refine our diagnosis, but in a lot of tumors, we're still struggling to know what the best treatment is. And it's things like information like that that's going to really help us move forward with the specialty.
Another question by Erica is, when deciding what sequencing platform to use for these samples, what made you choose short read? Illumina. If you could go back, would you still choose short read or test the novel long read tech?
Excellent question, Erica. We're doing a lot of this at Great Ormond Street at the moment. So nanopore sequencing is the exciting thing we're doing. And the reason for that is it usually takes around 2 to 3 weeks to get these molecular tests back. And that's a long time to wait for a result. And it's frustrating for us. With nanopore sequencing and long read sequencing, we can potentially get the results back within an hour, which will be a remarkable change, which means the patient will be able to start on their treatment a lot quicker and hopefully will make a difference to the outcome for it as well. So there are different centres around the UK that are trialing this at the moment, but the aim is that every centre that has neurosurgery taking place should be able to use nanopore sequencing to provide that accurate molecular diagnosis.
Amazing. Thank you so much, Dr. Clarke. So because we're conscious of time, we'll end the question session there. For those of you whose questions are not answered, please feel free to email Dr. Clarke. So over to you, Dr. Saka.
Thank you. Thank you, Dr. Clarke, for the amazing presentation. And thank you, Joy. And thanks to all of you for your questions. We will send you short feedback survey by email. So please share your thoughts about the event via that. A couple of quick remarks from me. National Pathology Week is our annual celebration of pathology and runs all this week. So you may see events being held at your local hospital. As part of National Pathology Week, the college is hosting an online pathology quiz for undergraduates next week on Wednesday. Teams for up to 6 undergraduates anywhere in the world are invited to enter. You can scan the QR code to find out more about it and book your place. A recording of this webinar will be uploaded to our YouTube channel shortly, and we will circulate the link. You can watch previous careers webinars on our YouTube channel. Just scan the QR code here. And that brings us to the end of the webinar today. Thank you all. Thank you for the speaker, Dr. Clarke, Joy, the host, and team at the Royal College, Penny and Demelza, and thank you for listening. Bye-bye.